So first, I would like to thank you for the invitation to deliver the lecture in this conference. So, this will be the title of my talk today: NanoBiomaterials for the therapy of experimental inflammatory arthritis. And I am Dr. Rehan Khan, working as a scientist D at Institute of Nanoscience and Technology. So first, I would like to tell you about our institute. It's the only dedicated institute in India, which is primarily working on the field of nanoscience and technology. Here, the every expertise, every background of the scientist is from physics, chemistry, and biology. They all are working together on the one aspect, that is nanoscience and technology. And this is situated in Mohali under this institute is established under the umbrella of Nano Mission by DST, Ministry of Science and Technology. And before going into the detail of my talk, first I would like to tell you my areas of research. I am primarily working on the development of nanomedicine or drug delivery system for the management of uh, cancer and experimental colitis, experimental rheumatoid arthritis. Apart from this, I'm also working on the aspect of nanotoxicology, because we are developing engineered nanomaterials and new nanomaterials. So their fate is really unknown in the their fate is in terms of toxicity is really unknown. So we are also assessing the toxicity aspect of the
Sorry to
nanomaterials.
interrupt you, Dr.
Yes,
Rehan. Your
yes.
slides are not moving. It got stuck on the first slide.
Okay.
You have to just click on the slide which you want to show.
Okay. So this, I just want to show this slide. So can you see the slide? This areas of research.
Yes, sir.
Okay. Now it is moving.
It is on second slide only. Now it is on fourth slide.
Okay. Okay. So people have developed different types of drug delivery system for the management of different diseases. Liposome is the most commonly type of drug delivery system people have developed, and liposome or this lipid type of drug delivery system have reached the clinical trials, and now it's the in the market also, the nano formulation based on liposomes, and people have developed the micelles, nanocapsules, solid lipid nanoparticle, gold nanoparticle, and magnetic nanoparticles. So these are the type of drug delivery system people have developed. And these are the roots of drug delivery system. The roots of the drug delivery system is mainly oral and transdermal delivery. People have developed the patches in the form of for the transdermal drug delivery system. And for pulmonary delivery system, people have developed some nanoparticle like PLGA-based nanoparticle for the pulmonary or through the inhalation system. And for the nose, through the nose, it's like for the brain targeting delivery, people have using the nose type of inhalation system, and ocular type of drug delivery system people have used, and the parenteral. Parenteral is like a kind of intramuscular, subcutaneous, and intravenous type of drug delivery system. So these are the some background of the drug delivery system, and now I'm going to switch to my um, experimental details. So first, I would like to tell you about our first study, dual drug encapsulated modified polymeric coated gelatin nanoparticle, which attenuated the experimental rheumatoid arthritis. This paper has already been published in Carbohydrate Polymers. So before going into the results, first I would like to tell you the background of what is rheumatoid arthritis. Rheumatoid arthritis is a chronic autoimmune disease which is characterized by the severe inflammatory pain in joints. It is the most prevalent type of autoimmune disease among all the autoimmune diseases, and there is no perfect cure is available for this disease. Only clinicians are using the medicine for the symptomatic relief. And the hallmark is like a degradation of bone due to destruction of articular cartilage, inflammation of joints, inflammation and thickening of joint capsules. To better understand the rheumatoid arthritis in the form of stages, first you see that it's a healthy joint. As you can see here, this is a healthy joint. It's like intact bone and cartilage. And synovial membrane. Everything is very healthy. And the first stage of the disease is the synovitis. Synovitis is the inflammation of the synovial membrane, which leads to the thickening in the due to the inflammation. And the second stage is the pannus formation. Pannus formation is a kind of like extra outgrowth which form in the joint space, and it is like compressing the nerve also. That's why the some patients reported the severe pain in due to the arthritis condition, due to the rheumatoid arthritis condition, because of this pannus outgrowth. This pannus outgrowth is a kind of hyperproliferation in the synovial cell that lead to the formation of this pannus. And the third stage is fibrous ankylosis. Fibrous ankylosis is a kind of like a third stage. It is like a joint invaded by fibrous connective tissue. As you can see here, the cartilage is almost degraded, and this and in the last stage, as you can see here, bony ankylosis. This is the, like both the bones of like upper joint and lower joint, both the bones fused together, and the cartilage is completely destroyed in this stage, and this will lead to the disability of the patient, as like both the bones fused together, the bone joint are stiffed, and they cannot like flexibly move the joint. This lead to the disability in the patient. And in the other term, like again, to understand the stages of rheumatoid arthritis, the stage one, the body mistakenly attacks its own joint tissue, and in stage two, the body makes the antibodies, and the joint start up swelling. And start in stage three, the joint start becoming bent and deformed. The finger become crooked. As you can see here, in most of the like RA patient, the joint become bent. Sometimes they are unable to move their fingers or fingers of the hand, or like joints, and these like result in the misshapen joint can, and these like misshapen joint can press the nerves and cause the nerve pain as well. In stage 4, if not treated, the disease will progress to the last stage, in which there is no joint remaining at all, and the joint is essentially fused together. And this is the probable mechanism people have reported about the disease development, and the autoimmune trigger that lead to the activation of T cells. At the T cells, you can see here, T cells will lead to the activation of Th17 cells, and these Th17 cells secrete the IL-17 cytokines, and the T cells also activate the Th cells, and these Th cells activate the macrophages, and these macrophages also secrete this IL-17 cytokines, and these cytokines regulate the level of other key inflammatory cytokines, that is TNF alpha and IL-1 beta. And on the other hand, the T helper cells also activate the B cells, and these B cells activate the plasma cells, and these plasma cells secrete the autoantibodies, and these autoantibodies are responsible for the cartilage destruction. These autoantibodies are secreted by the chondrocyte, and they ultimately lead to the cartilage destruction. And these cytokines, TNF alpha and IL-1 beta, these are the like master regulator of the inflammation, and these cytokines like stimulate the synovial fibroblasts, and these synovial fibroblasts, as we well reported, that these are the responsible for the release of various degradative enzymes, mainly MMP2, MMP9, and these MMPs have osteoclastic nature. Osteoclastic nature, they have like damage the bone and the cartilage. They are mainly responsible for the destruction of bone erosion and cartilage destruction. On the other hand, people have also reported that neutrophils, which are the inflammatory cells, like these neutrophils degranulate and release the human neutrophil elastase enzyme, and these elastase enzyme is also responsible for the cartilage destruction. These are some of the mechanisms people have reported. But in our study, the aim of our study, we have used two different type of drugs, that is glyceric acid and budesonide. Budesonide is a well known, and it is already in the market, and it is known as like an SAID, like it is a corticosteroid, and it is like used for the management of this rheumatoid arthritis and ulcerative colitis. And the other is the glyceric acid. The glyceric acid is a natural compound which is a potent COX-2 inhibitor, and it like reported to have potent anti-inflammatory effect. And this glyceric acid is isolated from the Glycyrrhiza glabra plant, and it is like commonly known as licorice plant. And this glyceric acid is very effective in suppressing the inflammation, and it is already used in Japan for the management of viral hepatitis. So, and therefore, we have chosen these different drugs to see the effects, combination effects of these two different drugs. And the problem with the glyceric acid is water soluble molecule, but it has very high first pass metabolism, and therefore the bioavailability of this drug is very low. On the other hand, the limitation of the budesonide is also it has very low bioavailability. Therefore, we have designed this core shell nanocarrier for the encapsulation of both the drug in a single system. So this is the our schematic. First, we have chosen the polycaprolactone, and we have grafted the polycaprolactone with the aminocellulose. And first, aminocellulose we have synthesized with the help of cellulose. First, we converted the cellulose to the tricyl cellulose, and then we finally formed the aminocellulose that we have already reported in some other paper published in Biomicromolecules. And then we have chosen this aminocellulose to graft this to the polycaprolactone and to form the PCL-AC. The PCL stands for polycaprolactone, and AC stands for the aminocellulose. And we finally got this product, PCL-AC. And then after this, we separately synthesize the gelatin nanoparticle, and gelatin nanoparticle we synthesize with the help of precipitation method, and we have encapsulated glyceric acid drug into this gelatin nanoparticle. We have chosen gelatin because this is have the compatible nature, like both have the hydrophilic in nature and water soluble material. Glyceric acid and gelatin both are water soluble material, and therefore we have chosen gelatin to develop gelatin nanoparticle for this type of drug. And finally we got this glyceric acid encapsulated gelatin nanoparticle. And then what we did, we add this PCL-AC into this gelatin loaded nanoparticle, and we added PCL-AC and both budesonide into this in the solution of this glyceric acid loaded gelatin nanoparticle. And finally, we form this type of core shell nanostructure. As you can see here, the yellow internal core is the gelatin nanoparticle which contains the glyceric acid drug, and the layer is of PCL-AC, that is polycaprolactone, aminocellulose grafted polycaprolactone, and this contains the budesonide drug. And this polycaprolactone PCL-AC is the hydrophobic in nature, therefore it is and budesonide is also hydrophobic in nature, and budesonide will stay in the outer layer of this molecule. And next we have characterized our PCL-AC. First, we have characterized the PCL and AC. We found the characteristic peaks are present in the NMR, and we have confirmed the formation of this PCL-AC with the help of FTIR also, and the characteristic peaks of amide stretching at 3430 are present, and the amide bending 1663 are present in the this PCL-AC, which confirms that our conjugate of PCL-AC has successfully formed. And then we have further characterized with the help of XRD. And XRD, as you can see here, the amorphous hump of the aminocellulose, and is present in the PCL-AC. As you can see here, the amorphous hump of this is present in this, and the sharp characteristic peaks of PCL are also present in the PCL-AC, which further confirms that our PCL-AC have been successfully formed. Then we have characterized with the help of gel permeation chromatography to check whether the PCL-AC has been successfully formed or not. The molecular weight of the PCL is 135,000 Dalton, and then after the conjugation of aminocellulose, the molecular weight is reduced to 34,000. Why it has happened? Because the polycaprolactone is a long chain of polymer, and when AC will graft, the long chain of polymer will break into fragment, and AC is grafted on this. That is why the overall molecular weight of the PCL-AC is reduced as compared to the PCL, because it is long chain fragment into small pieces, that and the aminocellulose is grafted onto that. So that side, they further confirm that the PCLAC has been formed successfully. Then we have characterized our nanoparticle with the help of dynamic light scattering, and we first perform the size hydrodynamic diameter analysis with the help of DLS. First, we perform the diameter of the blank gelatin nanoparticles. The size of the blank gelatin nanoparticle was 138 nanometer and the PDI was 0.12. And after like a coating of the PCLAC gelatin nanoparticle, the size increased from 138 to 187. Like it is expected, like after the coating of PCLAC, the size increased, which also confirms that PCLAC has been formed. PCL has been grafted onto the gelatin nanoparticle. And then we calculate the theta potential also, measure the theta potential at 11.2 millivolt. And then we have finally perform the DLS study of the final our dual drug loaded PCLAC gelatin nanoparticle. Dual drug means both the budesonide drug as well as the glyceric acid drug is encapsulated into the PCLAC gelatin nanoparticle. And the size or hydrodynamic diameter of the nanoparticle was around 204 nanometer, and the PDI was 0.35, and the theta potential was 20.3, which like shows the satisfactory results to move further ahead. And then we perform the different microscopic technique to see the morphology and size of the nanoparticle. As you can see here in the TEM analysis, as you can see the particle size and the internal core, as you can see here, internal core is 135 nanometer, and the exterior like outer PCLAC layer is about 31 nanometer. In the TEM analysis, we have like clearly seen that our nanoparticle is of core shell structure. We further characterize with the help of SEM and FESEM, and then we perform the release pattern study. And the release pattern study is very crucial because after encapsulating the drug into the nanoparticle, it should release, the nanoparticle should release the drug in the physiological conditions. If it is not able to release the drug in the physiological condition, or it's like release immediately, then the nanoparticle or the drug delivery system is of no use. It shows up personally. Then it's like, therefore, we have performed the release study in using the dialysis bag method, and then we perform the release pattern of the GA glyceric acid as well as the budesonide, and both the drugs show the controlled release manner, and like most of the drug release in about 2.5 days. In 90% of the drug release in about 2.5 days. That's like which like shows that it is a controlled and sustained release behavior of the nanoparticle. And then we have performed the cytocompatibility study, and then we have used the HTERT cell lines, which is the human skin fibroblast cell lines, which is of like non-cancerous cells, and we have taken our blank nanoparticle. Blank means without any drug. We have chosen this blank nanoparticle to see the cytocompatibility of the carrier system only. So when we have performed the MTT assay, and we perform time-dependent study, 24-hour and 48-hour in the dose-dependent manner, and we found that up to the 400 microgram per ml at both the time point, our like nanocarrier did not show any significant cytotoxic effect. So it means like our like nanoparticle up to 200 microgram per ml is completely safe, and we can use it for the cell-based study. And then after performing the in vitro study, we have moved to the in vivo study, and we have developed the rheumatoid arthritis model. We have developed this model with the help of collagen-induced arthritis model. We have used collagen, type 2 collagen, and we have developed the emulsion of the this type 2 collagen, and by mixing the fluents, complete adjuvant, and we have injected into the tail, at the base of the tail, 1 centimeter away from the body. We have injected at the tail of the base, and then we have performed as you can see here, the very prominent erythema and edema was found in the arthritis group. The group 1 is control, and the group 2 is, as you can see here, CIA group. This is collagen-induced arthritis group. Group 3 is the CIA, which is treated with dual drug, only like a free form of drug. And then CIA, DDL nanoparticle means it is like dual drug loaded nanoparticle. And this one is the last group is the blank nanoparticle. In the healthy animal, we have injected our only blank nanoparticle without drug, just to see the effect of the carrier system, whether our carrier system showing any adverse effects or any protective effects. So therefore, we have injected into the rat only blank nanoparticle. And as you can see here, the prominent swelling and erythema and edema in the arthritis group. But after the treatment with the dual drug nanoparticle, as you can see here in the green circle, the paw is almost like very reduced swelling and reduced redness in the paw, at like more looks similarly to the control group. And as you can see here, the disease activity change in paw diameter after the treatment with the dual drug loaded nanoparticle. The mean change in the paw diameter reduced significantly after the treatment as compared to the arthritis group. These are some of the staining methods we have used. The first one is the Elsian blue neutral staining. This Elsian blue dye is mainly stains proteoglycans, which are one of the composed of like cartilage matrix, which like shows that it is the presence of the cartilage. As you can see here, this blue color is prominently present in the healthy joints, but in the arthritis joint, as you can see here, the this blue color means proteoglycan content is reduced drastically in the arthritis group. But after the treatment with the dual drug nanoparticle, this blue color is prominently present. It means that it is the our dual drug nanoparticle. It shows the cartilage protective effects. It like restore the proteoglycan content, which proteoglycan content is one of the part of the cartilage. So this is the our nanoparticle shows the uh, cartilage protective effect. And then we have performed the H&E staining and to see the histological alteration in the different regions of the joint, that is synovium, cartilage, and bone. And these are the three main regions which are like primarily affected in the case of the disease. And then we have seen that in synovium, as you can see here, the synovium is a normal synovium, and the case of arthritis, as you can see here, multiple inflammatory cells are present in the synovium in the case of arthritis group. But after the treatment with our dual drug nanoparticle, this inflammation of infiltration of this inflammatory cells drastically reduced in group D as compared to the arthritis group. And similarly, in case of cartilage, as you can see, there is a normal histoarchitecture of the cartilage in the joint. And as you can see in this, in the arthritis group, the architecture is completely distorted in the arthritis group. And after the treatment with our dual drug nanoparticle, the distortion reduced markedly as compared to the CIA group or arthritis group. This again shows that our nanoformulation shows protective effect against the cartilage protective effect. And now the bone, as you can see, histoarchitecture of the bone is like it looks like this. See here in the healthy group A, and after the treatment, the arthritis group, the bone histoarchitecture is completely distorted. And but after the treatment with our nano formulation, the bone histoarchitecture restores markedly as compared to the CIA group. And lastly, the toluidine blue staining is we have performed as toluidine blue staining is like used to stain the mast cells. As mast cells are the one of the key inflammatory cells which play very crucial roles in the development of some diseases, and mast cell is one of the hallmark of the inflammation. As you can see here in the healthy joint, there is no mast cells are present, but in the arthritis group, as you can see here, this dot blue, violet blue color dots are the mast cells, and these mast cells are heavily infiltrated in the arthritis group. But after treatment with our nano formulation or dual drug loaded nanoparticle, this mast cell infiltration reduced drastically as compared to the CIA group, which further shows that our nano formulation has anti-inflammatory effects. Then we have further performed some immunohistochemical expression of some inflammatory biomarkers like IL-1 beta, interleukin 1 beta, COX-2, and I0. And we found that the expression of all these three key inflammatory markers has been increased in the arthritis group, but after the treatment with our nano formulation, the immunopositive cells for these particular biomarkers reduce drastically uh, in the dual drug loaded nanoparticle group. And then we have performed some biochemical assays to further establish our hypothesis of like it is like anti-inflammatory in nature. And we have performed that a neutrophil elastase. Neutrophil elastase is one of the hallmark as like neutrophils recruited at the site of inflammation, and they releases this enzyme, this neutrophil elastase, and we have measured this neutrophil elastase enzyme, and we found that the neutrophil elastase enzyme activity was very significantly higher in CIA group as compared to the control group. But after the treatment with our dual drug nanoparticle, the activity of this enzyme reduced drastically as compared to the CIA group. It is reduced significantly as compared to CIA group. And nitrite level also like shows the anti-inflammatory effect, which is the indirect measurement of the nitric oxide level, which is also one of the like biomarker of the inflammation. And we found that in the CIA group, the nitrite level increased significantly in the CIA group as compared to the control group. But after our like dual drug nanoparticle, the level of this nitrite reduced significantly as compared to the CIA group. And then finally, we have measured the TNF alpha, and we found that the TNF alpha in the CIA group increased significantly as compared to the control group. But after the dual drug nanoparticle, the TNF alpha was reduced significantly as compared to the CIA group. And this is the plasma CRP, which is the clinical C-reactive protein, which is the clinical biomarker for the inflammation. It's like widely used in the clinic to establish the inflammation. And we found that the CIA group, the inflammation, the plasma CRP is increased significantly in the CIA group as compared to control. But after our treatment with our dual drug nanoparticle, the level of this plasma CRP reduced significantly as compared to CIA group. And finally, we have performed the histological study for the blank nanoparticle, and we have primarily chosen two vital organs, liver and kidney, because these two organs are mainly responsible for the biotransformation of any material injected or ingested. Suppose if we have taken any material through oral route or through, like, it goes to the extensively to the biotransformation in liver and then kidney. So therefore, we have chosen these two organs, and we have assessed the tissue compatibility of this our blank nanoparticle, and we found that there is no histological alteration in the histoarchitecture of liver and kidney as compared to the control group. It means that our blank nanoparticle is safe and did
Sorry
not.
to interrupt you.
Yes,
Sorry to interrupt you, Dr. Rahman.
yes.
Actually, we have more keynote speaker in line. So
Okay,
it's a
okay.
request to you. Please finish it as soon as possible, sir.
Okay, okay. And then the final thing, conclusion of this study, result of this study suggests that the dual drug-loaded core shell nanoparticle exhibits superior therapeutic properties over free or naive form of glyceric acid budesonide in experimental arthritis. The results may be assigned to their ability to significantly inhibit the inflammation. So one more study I'm going to just tell briefly about this study. We have developed this zinc gluconate study for the we have loaded this zinc gluconate into the chitosan nanoparticle for the rheumatoid arthritis uh, management, and then we have chosen the zinc gluconate as zinc is like essential biological trace element. It shows the zinc is anti-inflammatory effect, and it regulates the immune system and maintains bone health and low. The problem with the zinc gluconate is like it has very low absorption capacity. Zinc gluconate is already in the market and recommended for the use in the some childs also. That is why and zinc is responsible for the maintenance of the immune system. To overcome this challenge, low absorption capacity, we have developed this chitosan nanoparticle to encapsulate zinc gluconate, and we have delivered to the rat model, and we have performed some physical chemical characterization of the zinc gluconate, and found the final size of the zinc gluconate nanoparticle was around 106 nanometer, and we have performed some TEM and SM analysis, and we have performed these some studies of the drug release and cytocompatibility study of the our nanoparticle, and then we have performed this therapeutic efficacy study of the zinc gluconate nanoparticle. This zinc gluconate nanoparticle reduces the disease severity of arthritis as compared to the arthritis group. Like, and we have further performed many other studies to see the zinc level, like in serum, the zinc level increase. This is the free form of zinc without any nanoparticle, as you can see here, ZG, and this is the zinc gluconate cortisone nanoparticle. And we performed the serum zinc level, which measured after injecting into the rat, and we found that after encapsulating into the nanoparticle, the plasma serum level of zinc enhanced significantly, which shows that our we can overcome the limitation with the help of the drug delivery system. Therefore, the effectiveness may be increased because of this encapsulation. And we have performed some anti-inflammatory biomarker studies to further show that our nanoparticle has this anti-inflammatory effects, and we have performed this study, actually staining study. This is the joint heteroarchitectures in the arthritis group, as you can see here. The panel one is lower magnification image, and B dash is the higher magnification image of the same image. And we found that after the treatment with the zinc gluconate cortisone nanoparticle, the joint architecture is very intact, and it looks like the healthy one. And as you can see here in the CIA group, the joint is distorted. As you can see here, the outgrowth or disruption in the joints here, I've shown with the green arrows. But in the zinc gluconate nanoparticle treatment group, the joint is very smooth surface, and it looks like the healthy joint. And the result of this study that showed that zinc gluconate cortisone nanoparticle reduce the disease severity of arthritis and exhibit better superior therapeutic effect compared to the zinc. And these therapeutic effect may be due to anti-inflammatory effects of zinc gluconate cortisone nanoparticles. And lastly, I would like to thank to my institute, Institute of Nanoscience and Technology, Mohali, and my PhD student who worked very hard to do all these studies, and they have published in good journals. And lastly, I would like to thank the funding agency, DST, SERB. Thank you.